Right, the Scarce Bluetail at the cut, Sompting Brooks. Let's go back to August 2020. 2020, yes, 2020. I was going through Facebook, as you do. I couldn't go to Spain. I couldn't really go anywhere else. It was all full of lockdown and COVID and stuff. So leafing through Facebook, I happened to come across the Scarce Bluetail from West Sussex. And I thought, well, this must be a hoax. And then I saw who'd posted it. And it was a guy called Dave Sadler, who knows his stuff. And he's probably the top man in Sussex. So I thought, this is worth pursuing. So before you knew it, I was meeting Dave on site at the cut, Sompting Brooks. And I think before I start the talk, I need to make it very clear that the field work here has 90% of it has been done by Dave Sadler. A little bit backed up by me, but basically Dave has done all the work and I'm taking the credit. So let's progress. These are the various categories I've done. Let's just get on with them. Let's work our way through it. So first of all, the discovery. We are in southern England, of course, the Isle of Wight at the base there, and Sompting is there. Right at the foot of the South Downs, between Shoreham and Worthing. There's our site. Literally, it's an area of low-lying agricultural land, bordered to the north by the A27 and the Sussex Down and the South Downs, and to the south by the South Coast Railway. But literally, as you can see, it's fairly dull, normal agricultural land. Very low lying, about 4 meters above sea level max, and it has got a few drainage ditches which ran across it, most of which are simply completely overgrown. The area is pretty well devoid of dragonfly life, and it as an area, and most important point, it has no public access, or didn't have any public access until now. In the distance, you can see about the only thing that's famous in Sompting, which is the church. It's a very old church with a rather quaint roof. Anyway, that's all slightly irrelevant. Progressing, this is the Sompting estate viewed from above, literally the fields. And what the Sompting estate decided to do, because I think they got some money from various funds, was to do some rewilding. And literally this length of stream here, which as you can see is right next to an industrial estate, was rewilded by using money from the Rampion Fund. The Rampion Fund, you probably know, is the huge wind farm, which is offshore from Shoreham, and various other organizations advised. And this is what they did. This is the cut. And this is literally what they did. There was a little stream, I suppose you call it a stream, a drainage ditch that went right through the edge of the habitat. And it was previously completely overgrown, utterly useless. And they decided to excavate and clear and form this beautiful shallow basin with a shallow trench in the middle. And I must admit, I take my hat off to the digger driver, because the way he sculptured this beautiful length of stream was absolutely magnificent. There it is slightly later in the year, the shallow water developing. There were large patches of calitrichae or starwort, which were incredibly important for the beast. And then on the 19th of June 2020, a guy called Tom Foreman was sweep netting for invertebrates along the cut, and he found this, and he wasn't quite sure what it was. So he contacted Dave Sadler, who appeared and identified it as the Scarce Bluetail. In fact, Dave tells me he was slightly miffed at the time that somebody else was on his patch and had found a rarity, and I can understand what he was saying. So that is how it was discovered. The next thing we should look at is probably, I think let's consider the field work, what Dave decided to do. Now, instead of just looking at it and ticking it off and saying, fine, we've got a new species, Dave decided to do a rather systematic look at the species. He decided he would visit as often as possible throughout the summer when there were weather permitted. And I was drafted in to fill in the gaps when he was away. The most important thing with studying things like damselflies is the weather. It's not so vital that you have sun, but it is vital you don't have wind. Wind is the killer when studying small damselflies. The aim was to walk along the cut in both directions and to photograph all the individuals he could and to record all individual sightings. Obviously, you can't photograph everything because this damselfly is very small and one that is, shall we say, difficult and slightly capricious in the way that it behaves. So literally, you record everything and photograph everything. Spend at least 2 hours on site, of course. I used to spend about that long. Dave used to spend much longer. But that was how he decided to carry out the survey. Of course, analyze the data on the computer, and we'll come to that in a moment. Now, before we go into what we found, we need to look at what this site is. This is Dave Sadler in all his glory, next to the cut in August 2020. Lovely, low, shallow water, low vegetation. You're not visible there, but large areas of starwort, calitrichae, which were used by the females to oviposit. Now contrast that with the early part of 2021. The vegetation was growing very quickly, tall vegetation was appearing, open water was reducing very rapidly. There were attempts to clear some of the sites to keep the vegetation down. You can see here also these trees here are a species known as the black poplar, which I understand is extremely rare. Therefore, it was going to provide tree cover, which wouldn't be terribly helpful. There in August 2021, we have the habitat. This is, ladies and gentlemen, a habitat in transition, and you need to keep that in mind. Now, before again we find, I always like to ask myself a question, and one of the things that I always fascinates me is why? Why here? Now, this is a nice piece of habitat, yes, but what's special about it? Why on earth would Ischnura pumilio occur here? Bearing in mind, in order to put this in context, Ischnura pumilio, the nearest site for Ischnura pumilio is the New Forest, which is about 60 miles away from this site. So why would it be attractive here? Why doesn't it occur more places? So I've started to look at some of the places I know it well in Spain. This is the Bolsa del Pinar, which is one of my favorite ponds in Spain. And it has a similar profile, very shallow profile, that in the years 2012, 2013, and 2014 was completely bone dry. And only in 2015 and 2016 did the water come back to allow dragonflies to recolonize. And that's the Bolsa del Pinar in 2016. And this is a site, very common site, for a site of great importance for Ischnura pumilio. Now, the other sort of site you get a lot of pumilio in Spain are paddy fields. And this is a paddy field, a rice plantation near Jaén in central Spain in 2018. And you can see lovely open water, permanent water throughout the summer, which is a key point. And here we have even a photograph, folks, of Ischnura pumilio on a rice plant in this very paddy field in 2018. Now you're going to say to yourself, why on earth would this be important? Well, the reason is temperature, water temperature. In the cuts, over the periods of August to September 2020, we had an average temperature of this lovely shallow water of about 24 degrees. In the cut in May to August 2021, it was even warmer, 26. Look at some of the maximum temperatures. If you look at comparison sites quite close to the area, even in low-lying areas, the similar pond, most of the pond areas, and of course they're that much deeper, they have a much lower water temperature.
Yeah, they're 10 minutes.
Hello? Oh, sorry. Anyway, the cut basically 52% warmer than comparison habitats. We're looking at a lovely warm bath, which is fine for a damselfly that breeds in this type of habitat. How the pumilio finds such warm sites, I'm afraid I couldn't answer. But anyway, now having worked out why it's there, let's look at what we found. First of all, the males. And this is where Dave's technique of photographing everything came into its own. Dave ascertained that the male Ischnura pumilio has got incredible variability in the segments 7 to 10. The various areas marked here, A, B, C, D, blah, blah, are all very variable. Not only are they variable in color, they're also often asymmetrical. There are shapes in this area B here, which are asymmetrical. These markings D are very important because they are hugely variable. And what we found is you could identify individual males from their markings. And interestingly, there appear to be different males. The markings do not repeat from year to year. It would appear that the markings of 2020 were different from 2021. So you could identify individual males. This one is probably one of the most extreme examples. If you read Grand and Buda, the dragonflies of France, they state that you can get Ischnura pumilio that has a completely black mark here. So there's no blue mark at all, which we didn't find. But look, this one, how much black there is on the ninth segment. Moving on, this one here with much smaller black, and the one at the last one is this one. And the most interesting thing was that in 2020 and in 2021, towards the latter part of the season, this completely clear example was by far the most common. Earlier in the season, these variable different patterns were common, but in the latter part of the season, this one, the clear marking, was more common. Anyway, the overall observations in 2020, as you see, between 2020 and 2021, the actual observations of males was fairly similar. As you can see, different males, we identified 60 in 2020, 73 in 2021. Different males identified from their color patterning. So the males, basically, they were about the same. So we move on to the females. Now, the female of Ischnura pumilio is unique2 with one other species in the US, in that it has, Ischnuras usually can have between 2 and 3 female forms. And the 3 female forms are different between them in a juvenile stage. The Ischnura pumilio has 2 female forms. This one, the Aurantiarca, the juvenile. This one, the gynochrome, the one the proper female, and this one, which is the blue female, the androchrome, as you would call it. What makes them different from other Ischnuras is both the androchrome and the gynochrome both stem from the original Aurantiarca morph, which is the only other Ischnura that does that is one in the US called Ischnura kellicotti. So we have here, these are our female observations. 43 female observations in 2020, but only 17 in 2021. we had 7 androchrones in 2020, none in 2021. All we had in 2021 was 17 gynochromes. In other words, in 2020, we had lots of females. In 2021, we had a greatly reducing population. This is shown the copulas, and here is a female. The juvenile is often found in copulation. In fact, there are Ischnura species where they only copulate when they're young. They will not copulate when they get older. I won't get onto that. We'll be here all night. Yeah, as you can see here from the copulas, well, we saw about the same from 2020, 2021. Far more in the earlier stage than 2021 and fewer. What was more interesting is in the oviposition. Most oviposition was taking place in the calyptricae, the starwort, and there is a blue female. Now, you might think that the blue female here is ovipositing in tandem. This is not the case. What was happening generally was that the female, this one particularly, this blue one, was ovipositing, and the males would come in and attempt to mate with them by clasping them. In most cases, the female completely ignored the male, and the female carried on. I think there was one or two cases that Dave recorded where the female did actually go away with the male. One case that Dave photographed, and this, I have studied Ischnuras in Spain and in the Middle East, and I've never seen this. This is a female Ischnura ovipositing underwater, and I've never seen that before, and I don't think it's commonly recorded. The most interesting thing about oviposition was look at the difference. In 2020, we had 20 cases of oviposition, and almost later in the year. And in 2021, we had only 2. So it looks like the females were deserting a sinking ship. So that's what we found. But what have we learned from this? This is the Latchmore flushes. I was listening to Magnus this morning, holding forth about how wonderful, how he loves bogs. Well, the Latchmore flushes in the New Forest are some of the finest, beautiful, beautiful habitat. Note the temperature, 27 degrees mid-afternoon on the 14th of July. Magnificent shallow water habitat, a permanent site in Britain where you're almost guaranteed to find Ischnura pumilio. Now, Dave Thompson, legend amongst dragonfly researchers, and I think it's Kate Allen, back in 2010, they did a lot of work on the Latchmore flushes. I think it was about 8 weeks in June and July. And they also studied at the Red River in Cornwall, and they did a mark recapture venture. And they were there to establish the overall population size and dispersal. And their findings were that Ischnura pumilio is the most sedentary odonate studied in the UK to date, and that movements over 150 meters are rare. Now, contrast that with Grand and Budo, which is the dragonflies of France, highly transient species, colonizing new habitats, surviving only a few years. Now, this doesn't seem to match up. Can we learn anything from the cut of whether there's a difference? So I've now got a number of headings. Whether I'll get through them all depends on the time, but let's see how we go. Dispersal. Now, when Dave, this is Allen and Thompson at Latchmore, or they collected 2,000 plus insects. Now, the only people I know who've been to Latchmore will say, yes, you can find Ischnura pumilio, but you never see it in large numbers. Well, the team in 2005 captured a total of 2,000 insects over the 8-week period, 45% of which were female. 35% were recaptured. That's interesting. On the Red River, 250 captured insects, 36% female, 37% on recapture. Now compare this with what we had. Now I'm not using the recapture data because it's not compatible with what we did or what Dave Sadler did. We basically, I'm looking here only at what our observations were, comparing with what they captured. 45% at Latchmore, 37% in Red River. In the cut, in 2020, we had 17%, and in 2021, only 7%. All these observations, by the way, are based on adult, mature insects, which is exactly what Allen and Thompson did. 20 ovipositions in 2020 and only 2 in 2021. A great reduction of vastly smaller number of observations. 2020 males, 2021 males, hardly any different in terms of male population over the overall summer. Females quite different. Now we come to the Chelmick, some of the Chelmick theories, which you may or may not agree with. Permanent habitats, minimum dispersal, they don't move. Fair enough. Certainly that's exactly what Allen and Thompson ascertained. But we're talking about a transient habitat, and this is where you need to disperse. And it looks to us as though the females leave the changing habitat even before they have mated, because there were very few examples of mating in 2021. It looks as though the females emerge and decide this habitat is changing, we are off. Now contrast that with the males. Now the males stick around, because if you think about it, where are you going to get sex if you move? Your only chance of mating is going to be to stick around. Let's look at an example of a beast. This is an Ischnura that's found in the United States, very common along the whole of the eastern seaboard and going quite a long way inland. And that's the Orantiarca female, the juvenile female. Beautiful, beautiful creature. And there's an adult, which is rather more dull. Now, these beasts have been found breeding parthenogenically in the Azores. And I think to myself, well, maybe perhaps other species of Ischnura do this. Perhaps all Ischnuras, perhaps all damselflies, we don't know. Perhaps they decide that the habitat's not going to be up to it, they will move on. And they have an ability to mate, to lay their eggs without mating. Who knows? I've often wondered why, I've never understood why Ischnura hastata in the Azores, it breeds parthenogenically. And I've never worked out why somebody doesn't actually take some eggs or larvae from a colony in the US and see if they do it all the time. Because you can't tell me you suddenly get a population that goes to the Azores and they only breed parthenogenically. It must happen throughout the species, surely. Anyway, that's off the point. Male tail light. Right, as you've seen this, these are the two that you get early in the season, and the one later in the season is this one without the marking. So I thought I'd look at all the photographs I've taken of Ischnura pumilio over the years in Spain from my Spanish database. It's a total of 17 years of work from 700 sites, of which a few sites, about I think 6% of the sites. Remember, in Spain, I've never seen Ischnura pumilio on a permanent site. They appear not to be permanent sites. It's always a species that is in transitional habitats that come and go. And here you can see the marked tail light occurs throughout the year, but the increase is in the unmarked tail light as the year moves on. This is week numbers along the bottom, folks, and that's numbers. So more observations later in the year with a completely clear tail light. Do the males have such diverse tail lights in permanent habitats? Has anybody studied it? Has anybody done some not population study work, as was done by Allen and Thompson? What about studies of the particular males and whether these markings occur at habitats such as Latchmore? Why? Why would you need these markings on the end? Why do they reduce as the season progresses? Is it that the males need special marks to attract a smaller number of females that emerge early and that later in the season you don't? I leave you with that thought. Now, the famous blue female, the mystery of the blue female. I must admit, folks, when I first saw this in Spain some years ago, I hadn't the remotest idea what I was looking at. I hadn't got a clue. And I had to send my photographs to various people who eventually put me right and showed me that this was the blue female. It's the first time I'd ever seen it. It's a quite distinctive, they call it an androchrome, but to me, it doesn't really look like an androchrome at all. It just looks like the blue female. Anyway, Cam et al., 2010 first record, 2012 Norfolk Coast second record. It also states widely known in Europe and that blue females are often associated with migration. Well, that could be the case. Grant and Boudot slightly dispute this when they say the blue female is rare in France. So what did I do? I thought, let's check back at what Chelmick found on his Spanish database. 6% of all sites that I worked in Spain, so over 700 sites, I found Ischnura pumilio. All of these sites were transient. They were like paddy fields, temporary pools, etc., etc. Interestingly, androchrome females from my photographs, 13%. At the cut, we had 16%. And in fact, they're about the same. If you think 18% gynochrome, 13%, it's not unusual in Spain, in transitional sites. Hypothesis. Well, here we go. Permanent sites do not have blue females. Well, I have spoken to 2 people who have observed Ischnura pumilio down at Latchmore, and they tell me that they've never seen the blue female there. It will be worth more study. Does it occur? Transient sites, blue female. Why would you have, what's the advantage of having a blue female on a transient habitat? I don't know. It's worth thinking. Because the blue female is associated with migration, does it mean that the colonization came from across the channel? Yet another question for you folks. Now, Dave Sadler, because of the looking at the males, you can tell from the males, individual males, from his photographs. So obviously you can work out the length of life of individuals because all these males, and you can see how often that you photographed it. Now, when Allen and Thompson, of course, from recapture, you can do similar thing. You just recapture the data and see how long they live. Well, at Latchmore, all mature insects is not from emergence. This is the length of life of a mature insect. Latchmore, 8.3 days and 8.1 for females. The Red River, 6.8 days and 5.8 for females. And Dave, at the cut, most males he photographed live for up to 14 days, at least 26 days. Obviously, we have no data for females, but there's another theory here, folks. Perhaps in permanent sites with stable populations, individuals are short-lived. Maybe they lay a lot. Maybe the larval populations continue to spread over longer periods. They have larger volatilism. We don't know. Maybe for transient sites, the unstable population, the individuals have to live longer simply because they need to migrate and to find new habitat. Who knows? More questions, folks. Volatilism. How many generations per year? If you read Grand Budo, they say if you go right into the north, into where I think it does get into southern Sweden, there are one generation of larvae needs 2 years to complete. If you go more to the mid-levels, 1 to 2 generations, stated by Grand Boudin, I'm not quite sure what number of generations. So what I then did, again from Facebook, I found this wonderful guy, Steve Hewitt, who may be out there somewhere, who knows. He produced some data on Ischnura pumilio based upon BDS records and NBN of the total number of records and when they occurred throughout the year. So you can see it starts in May, goes to a peak in sort of June, early June-ish, and then declines down. This shows single generation. And in fact, Steve came on a paper he wrote some years ago about a site, I think it was in Sundon in Berkshire. He gives a very similar profile. 1 generation, literally May through to the end of May, June, and then completing life in August. So what this Steve Hewitt then did is he then progressed that to 2006 to 2021. And look how different it is, how much later the records are. And it certainly doesn't show a single generation profile anymore. And if you add to those records the cut, the ones in green, if you take all the records, it would show a definite 2-generational situation with a much bigger generation coming in August, going right through to September. So I thought again, well, let's see what the Spanish situation might be, because right in the south, according to Grand Budo, 2-3 generations. And I think that's probably right, because if you take the Steve Hewitt's analysis for 2006 to 2021, and you add Chelmick's Spanish data, the whole thing goes a long time. In fact, my latest record was in week 47. In actual fact, it was a teneral insect. So what can we draw from this? Almost certainly from permanent habitats, we have no idea. And I don't know that you could ever work it out, because I suspect the populations in permanent habitats, do they show generational interest? It doesn't seem to indicate that from Allen and Thompson's work that there is any, but we don't know. But certainly seems in transient habitats, mid-latitudes, at least 2 generations. And in the south, well, in my opinion, it probably flies all the year. All depends. If the habitat stays wet enough, it will fly all year. So there we are, folks. Now, that I think is how have we done? Not too bad we are. I think we've caught up beautifully. If you're still interested in this fascinating insect, folks, you'll be glad to know there's Mr. Sadler and Mr. Chelmick have written a paper which will appear in the Journal of the British Dragonfly Society this very year. I think it's coming out in the spring. And if you're still interested, you might like to look at Chelmick and Budo on Ischnura in the Middle East. And if you're still very interested, and I would recommend this paper, because this paper here gives the evolutionary history of color polymorphism in Ischnura. And well, you get the impression that the Ischnura genus are gradually trying to eliminate the need for males. And on that happy note, folks, that's my talk. Thank you very much.